Welcome to G Fun Facts Online. Imagine a world where you don't need a painful swab stuck up your nose to find out if you have the flu. What if, instead, you could just chew on a piece of gum? If it suddenly develops a strong herbal flavor, you've got your answer. This isn't science fiction. It's a revolutionary new diagnostic technology that could completely change how we detect and manage one of the world's most common infectious diseases. Today, we're taking a deep dive into the science of the chewable flu test, a technology that promises a literal taste of diagnosis. To really appreciate this breakthrough, let's quickly remember the current state of flu testing. For most of us, it means the dreaded nasopharyngeal swab. It's uncomfortable, invasive, and for kids, it can be downright distressing. After that unpleasantness, you face a trade-off. The most accurate tests, called PCR tests, are considered the gold standard. They can detect tiny amounts of the virus genetic material, but they're slow and expensive. They require a specialized lab and trained staff, meaning you wait hours or even days for a result. On the other end of the spectrum, you have the rapid antigen tests you can buy at a pharmacy. They're fast and convenient, giving you a result in about 15 minutes. The problem? Their reliability is a coin toss. They have a notoriously high rate of false negatives, with a sensitivity that can be as low as 50%. This means half the time you have the flu, the test might tell you you're fine, leading you to spread the virus unknowingly. This is the gap that scientists have been desperately trying to fill, a test that is as convenient as a rapid test, but as reliable as a PCR test. And the solution might be found in our own saliva. This new era of diagnostics is being pioneered by a team led by Professor Lorenz Meinl at the University of Würzburg in Germany. Their idea is brilliantly simple. They've designed a special molecule that acts as a sensor. When this sensor is put into a piece of gum or a lozenge, it remains completely tasteless. However, if the influenza virus is present in your saliva, the sensor gets broken apart by the virus, releasing a very distinct, bitter thyme-like flavor. The flavor comes from a molecule called thymol. So the process is straightforward. You chew the gum, and if you taste thyme, you likely have the flu. If it remains flavorless, you're in the clear. It effectively turns your own tongue into a highly sensitive, readily available flu detector. The initial lab results are incredibly promising. When researchers tested the sensor in vials containing saliva from people confirmed to have the flu, it successfully released the thymol flavor in under 30 minutes. With these results, the team is now preparing for human clinical trials, which they hope to begin in about 2 years. Now, how does this actually work? What's the science behind this flavor-releasing trick? The secret lies in targeting a crucial piece of the influenza virus machinery, an enzyme called neuraminidase. You've probably heard of flu strains like H1N1 or H3N2. The N in those names stands for neuraminidase. It's that important. To understand its role, let's look at the virus life cycle. First, the flu virus uses another protein on its surface, called hemagglutinin, or H, to latch onto our cells. It's like a key fitting into a lock on the cell's surface. Once attached, the virus gets inside, hijacks the cell's machinery, and makes thousands of copies of itself. These new virus particles then travel to the cell's surface to escape and infect other cells. But there's a problem for the virus. They are still stuck, tethered to the very cell they just came from, by that same hemagglutinin protein. They can't break free. This is where neuraminidase comes in. It acts like a pair of molecular scissors. Its one job is to snip the connection holding the new virus particles to the cell, setting them free to spread throughout your body. Without functional neuraminidase, the infection would be contained to just a few cells and fizzle out. This critical function makes neuraminidase an excellent target. In fact, our best antiviral drugs, like Tamiflu, are neuraminidase inhibitors. They work by jamming these molecular scissors, trapping the virus, and stopping its spread. Since neuraminidase is so essential and is active from the very beginning of an infection, it's the perfect marker for a diagnostic test. So, the German research team built a molecular sensor designed specifically for these viral scissors. Think of it as a flavor bomb with a very specific lock. The sensor consists of two parts, a flavor molecule, thymol, and a chemical lock that the neuraminidase enzyme recognizes. This lock is a synthetic version of the sialic acid that neuraminidase naturally cuts. The thymol is chemically bonded to this lock in a way that makes it completely tasteless. It's there, but your taste buds can't detect it. But when you chew the gum and the flu virus is in your saliva, its neuraminidase scissors get to work. They see the synthetic lock, think it's their natural target, and snip it. This cleavage breaks the bond holding the thymol, releasing the bitter, herbal flavor. The flavor is the signal that the viral scissors are present and active. But the researchers had to be extra clever. Our mouths are full of bacteria, and some of them produce enzymes that are similar to neuraminidase. To avoid a false positive from these common oral bacteria, the team made a tiny chemical tweak to their sensor. They modified the lock part of the molecule in a way that the viral neuraminidase can still recognize and cut it, but the bacterial enzymes can't. This makes the test highly specific to the influenza virus, which is crucial for any reliable diagnostic tool. The advantages of this chewable test over our current methods are immense. The most obvious one is comfort. It completely eliminates the need for an invasive nasal swab, making it a painless and stress-free experience. This is a game changer, especially for testing children, the elderly, or anyone with a phobia of the swab. But the benefits go far beyond comfort. This test has the potential to offer the best of both worlds, the speed and convenience of a rapid antigen test, with accuracy that could approach that of a PCR test. One of the most significant potential advantages is pre-symptomatic detection. People with the flu are often contagious for a day or two before they even start feeling sick. This is a major reason why the flu spreads so easily. Current tests, especially rapid ones, often fail during this early period because the amount of virus, or viral load, is still low. A highly sensitive taste-based test could pick up the infection in this pre-symptomatic window. Imagine you're feeling fine, but you chew a piece of this diagnostic gum just in case, and it turns bitter. You now know you're infected and contagious. You can isolate yourself, preventing spread to your family, coworkers, and community. This early detection also means you can start antiviral treatment sooner, which is most effective when taken within the first 48 hours of infection, potentially shortening the duration and severity of your illness. The use of saliva itself is a huge step forward for diagnostics. For years, blood has been the gold standard, but saliva is rapidly emerging as the diagnostic fluid of the future. It's incredibly easy to collect, requires no needles or trained medical staff, and can be done anywhere. More importantly, saliva is a rich source of biological information. It contains a diagnostic alphabet of proteins, hormones, enzymes, and genetic material from our own bodies and from any pathogens present. For respiratory viruses like influenza, several studies have shown that saliva tests can be just as, if not more, effective than nasal swabs. Some research has found over 90% agreement in detecting viruses like coronaviruses when comparing saliva to nasopharyngeal specimens. This is because the virus actively replicates in the salivary glands and is present in the fluid we swallow and breathe out. Of course, saliva testing isn't without its challenges. The composition of our saliva can change based on the time of day, our diet, or even our age. These variations could potentially affect a test's reliability. Therefore, a major part of developing this technology will be standardizing the collection and analysis methods to ensure the results are always consistent and accurate. Despite these hurdles, the field of saliva omics is advancing quickly, making tests like this chewable sensor more feasible than ever. So when can we expect to see this in our medicine cabinets? The road from a lab discovery to a consumer product is long and full of challenges. The first major step is the human clinical trials, which, as mentioned, are about 2 years away. These trials are essential to prove that the test is safe and that it works reliably in real people, not just in a test tube. The researchers will need to confirm its sensitivity and specificity and ensure that users can reliably detect the flavor change. After successful trials, the test must undergo rigorous evaluation by regulatory bodies like the FDA in the United States. This process is designed to protect public health and ensures any new diagnostic test is effective and safe for public use. Simultaneously, the challenge of manufacturing needs to be solved. How do you go from making a few sensors in a lab to producing millions of units cheaply and consistently? To tackle this, the research team is already working with a startup company called Flareon Biotech to prepare the technology for mass production. They estimate this entire process, from now until it's a market-ready product, could take about 4 years. Finally, there are the ethical considerations that come with any direct-to-consumer at-home test. Companies must be transparent about the test's accuracy and limitations. Clear instructions are needed to prevent users from misinterpreting the results. And, crucially, data privacy must be paramount. If these tests connect to an app or database, users need to be confident that their personal health information is secure. Looking beyond this specific flu test, this technology is a window into the future of medicine. It's part of a massive shift towards what's called point-of-care testing, moving diagnostics out of centralized labs and directly into patients' hands. The COVID-19 pandemic showed us just how critical rapid, accessible, at-home testing is for managing a public health crisis. The beauty of this chewable sensor platform is its flexibility. The lock part of the sensor, the sugar building block, is the part that's specific to the flu virus. By changing this component, scientists could theoretically redesign the sensor to detect other pathogens. Imagine a future where you could have a panel of taste-based tests for strep throat, COVID-19, RSV, or other viruses. The economic impact would be substantial. A study estimated the annual cost of seasonal flu in the U.S. at over $11 billion, much of it from lost work productivity. By enabling earlier diagnoses, isolation, and treatment, this technology could significantly reduce that economic burden and, more importantly, save lives. This represents a return to a more sensory-based approach to medicine, but one supercharged with 21st-century biotechnology. In conclusion, while there are still hurdles to clear, the chewable flu test is a landmark innovation. It promises a future where a simple, painless, and rapid home test can stop an illness in its tracks. It's a major step towards a more proactive and personalized healthcare system, a future where a simple taste of diagnosis could be the key to a healthier world.